All right, hello biology. This is the last video for the notes for Unit 5. Um, we took most of these yesterday, Monday, and we finished them up in third hour today, and in seventh hour we're going to finish them tomorrow because y'all are a little behind. Third hour is 3 slides farther than this on Monday, but I'm going to start here. That way this video will work for both classes. So we're going to start with transcription. So this is assuming you've already watched both of those Amoeba Sisters videos that were linked in the last video. Transcription is the part where you are actually scribing, writing out the RNA, writing out the message. That's scribing. Scribes write. Okay, so transcription is copying down the message from the DNA to the mRNA so it can be sent out of the nucleus to a ribosome. So transcription is that whole process of copying the DNA, making that mRNA strand, and that occurs in the nucleus. Okay. Why? Because we don't actually, or because all of the information that we need to make proteins is coded on that gene. So we'll make a quick copy of the DNA, then make a copy of the mRNA, and then send the mRNA off because we need to make a protein. So the enzyme RNA polymerase runs down and unzips a section of the DNA. It links the RNA nucleotides to the DNA strand to form the the mRNA molecule. Okay, and then it copies the entirety of one gene. It doesn't copy multiple genes; it is one gene. Okay, now remember, RNA doesn't use thymine; it has uracil instead. So every time there is an A in the DNA, normally we would attach a T to that. Since we're making an RNA copy, we use uracil U instead. Okay, translation then is where we actually change this message, which is written in RNA coding, into amino acid language so that we can read the protein off or make the protein. So translation is where we turn mRNA into a protein using RNA or using tRNA. And this happens on the ribosomes where the mRNA winds up after being copied. Purpose: to make a protein. Every strand of mRNA is coded to make one specific protein. So the mRNA leaves the nucleus, enters the cytoplasm. So we have our copy. This is translation or transcription. We make our copy, send it out of the nucleus to the ribosome, comes in this side, and for every codon, which we'll talk about here in a sec, we add one amino acid. So each of these is an amino acid. A bunch of amino acids together is a protein. Now, the way we attach the amino acids is by using tRNA. So mRNA can be broken down into 3-letter segments called a codon. Okay, a codon then has, or every codon then has an anticodon, which is tRNA. So the anticodon is going to be attached or part of the tRNA so that the tRNA will bind to the mRNA, and with the tRNA comes an amino acid. So there's an amino acid attached to the tRNA, it comes in, attaches to the mRNA. That tRNA amino acid then links to whatever amino acid is already there from the previous one. Then the tRNA breaks away from the amino acid and the messenger RNA, and it floats off to go bind to a new amino acid so that it can do that again. This process repeats until we hit the end of the mRNA, or what is called a stop codon, and then it just ends. At that point, we have a protein. The bonds that hold the amino acids together are called peptide bonds. It's a special kind of chemical bond that you will need to remember. And once the whole strand has been translated, the chain breaks off of the ribosome, and you have your protein all done. So the codon is a 3-nucleotide sequence. It codes for one specific amino acid. Multiple amino acid, or one amino acid can have multiple codons that it will match with. But every codon must pair with one specific amino acid. Okay? This is an example of a codon table. And I recommend that you pause this and take a picture of this if you want a color version of this chart. Because the one that's in the notes, if you scroll down a little bit, that one's not color-coded. So I'll leave that up to you. You can come back and take a picture of this. The way this works, let's say that our 3-letter codon is AUG. Okay? So we would start in the center with A. So we started here. We're going A. Okay? Now we have to go from A to U, because that's our second. So we go to here. Okay? Then the third codon is going to be the next here. That's G. So here. And G matches with this amino acid right here. MET is a 3-letter abbreviation for an amino acid. Now the one that's in your notes, the picture, if you scroll down, I was talking about earlier, that has the full name written out. This one just has these 3-letter bits. Notice down here, these two and this one are red, and they say stop. There are 3 different stop codons. So each of those will end a chain. That's how the RNA polymerase knows to stop making a copy. This is the end of the mRNA chain. And no tRNA will bind to it. This is a more classical version of a codon chart. I personally don't care for this one so much. I like the new one better. So the way this one works, let's go with the same codon, AUG. The first position is here, so we are in this row. Then the U comes from the second position. That's on the top, so it has to be here. And that overlaps in this area here. But you see we've still got 4 possibilities, so we go down to this end. We have UCAG. G is the one we want. It's on the bottom. So this would be our final answer. And again, you'll see that's MET. So the anticodon is the tRNA version of the codon. So if our codon was AUG, then this is the mRNA codon sequence. The tRNA sequence is going to be the complementary RNA strand. So A becomes U, U becomes A, and G becomes C. So this is the codon in the mRNA, and this is the anticodon, which is tRNA. Okay, and the last bit is the ribosome. Ribosomal RNA is where ribosomes are where protein synthesis actually takes place, at least the translation portion. So let's practice. If we have the DNA-based sequence AT, CG, CG, AT, CC, AA, T, GG, the mRNA copy of that, we'd have to use anywhere we would normally write a T, we're going to put a U. So remember, at group chat, ATCG, or GC. So A would normally be with T, so this is going to be a U. U is copied from the A when the messenger RNA is being copied. T would become A, G would become C, C would become G, then G, C, U, because we would put a T here, A, G, G, U, U, A, C, C. And I drifted up a little bit, but that's okay. And if we scroll down, that's what we see. So U, A, C, G, G, C, U, A, G, G, U, U, A, C, C. Now, the tRNA sequence is the complementary RNA. So it's a copy, but using RNA instead of DNA again. So again, we have U and A instead of T and A. So U would become A, then U, G, C, C, G, A, U, C, my brain stopped working, C, A, U, G, G. And again, A, U, G, C, C, G, A, U, C, C, A, U, G, G. Cool. I'm getting this. Now, from here we go to the amino acid sequence. Now, for this, we have to look at our codon chart. So if we switch to that real quick, actually, first, let's break this up into our, we've got our codons. This is very important. Your codon chart uses codons, not anticodons. So we need to use these sequences. So we need to look up the codons, or the amino acids, for UAC, GGC, UAG, GU, I wrote an A, hang on, GUU, and ACC. So if we go back, our first one was UAC, so UAC, that's TYR. Should write it down here, and I should use a different color. Where's my blue marker? So this is T. You cannot see that at all. Nope. So our first one is TYR, then GGC. GGC is going to be GLY, or glycine. UAG, UAG, that's a stop codon. So we're going to write in all caps, stop, because this codon is out of order. GUU, GUU, valine, VAL, and ACC, go ACC, that's THR. I don't know what that one stands for. So that would be tyrine, glycine, stop, valine, and whatever THR is. Now, if we go on, the way you would write that, oh, I was wrong. It's not tyrine, it's tyrosine, glycine, stop, valine, and threonine. Notice there's a hyphen between each. You must capitalize stop. Stop should be at the end most of the time, not in the middle. I kind of made this one up, and so it's somewhat nonsensical here. This is the bell work from Tuesday. So that's basically what we just did again. Also, I forgot to mention, worksheet 6 was practice over what we just did as well. So what we're going to go on to next is worksheet 7. Okay, gene is just that section of DNA. We've talked about that before, so I'll let you copy that down. Okay, these are the 20 amino acids that the human body needs. Now, if you pause this and look at this, you'll see that there are non-essential amino acids and essential amino acids. Non-essential amino acids, your body can produce from base elements. Essential amino acids, your body can't make. So you have to ingest those amino acids in order for your body to get them. You have to eat them. Okay? Proteins, organic molecules composed of amino acids, that's nothing new. Okay, and then the only difference between proteins is the amino acids that make them up. The number of amino acids, so a protein with 3 amino acids is going to be different from a protein with 4 amino acids, and the order that they're in. So if it's glycine, valine, leucine versus glycine, leucine, glycine, valine, that's obviously 2 different proteins. By the way, I made those up. They probably don't actually exist. Okay, mutations. Mutations are random changes in the nucleotide sequence within a gene. Now, this is a potential problem, and there are 3 ways or 3 things that we can really see mutations. Either a nucleotide gets added into the sequence, one is taken out of the sequence, so it's missing, or one gets changed for something that it shouldn't be. This happens very, very rarely, but it does happen. When it does, we call it a mutation. So frame shifting is when one is added or lost. So here, and I'll just kind of illustrate why this is a big deal. Here's a normal DNA strand. Okay? So our codons from our mRNA strand would be leucine, ALA, cysteine, and ILE. Okay? But if a G gets added, then it throws off everything after. So instead of the 4th codon, or instead of the 4th, 5th, and 6th being GCA, or the 2nd codon being GCA, our 2nd codon becomes CGC. Well, that codes for a different amino acid. And that means that every amino acid after that is also going to be different. And that means that the protein is not going to function the way it's supposed to because the amino acids are out of order. Because your DNA doesn't have a brain that can process and go, oh, that's not supposed to be there. Your brain or your DNA replication functions on the assumption that this is correct. And this is what causes some issues. So here's a good example. If we take the sentence where the sentence is the messenger RNA, and we remove a letter, so the fat cat ate the rat, that makes sense. But if we are missing the letter H, then instead of the fat cat ate the rat, our sentence becomes Tef atka ada tet her. Huh? That doesn't make any sense. Okay? Because that letter is missing, each codon is now a different sequence. And so it messes up the whole sentence. So the protein won't make any sense. It doesn't function the way it's supposed to. So these are some disorders and syndromes that we associate with deletions and insertions. And the other kind of mutation is a point mutation. Now this is when, rather than sticking something in that doesn't belong or taking something away, it's missing. So instead of, let's say, insertion, you're putting a puzzle together and you've got a piece that doesn't belong. Okay? Deletion, you're missing a piece of your puzzle. In point mutation, your one piece of your puzzle has been taken away and replaced with a piece from a different puzzle. Okay? So one nucleotide has been exchanged for a different base. So there are 3 different ways that this can be done. Now, there is a chance in a point mutation that it doesn't actually affect anything. Okay, because if you, let's take this one for example, if we had ATG and we swap it out for ATA, then it still codes for the same protein. But if either of the other two, if the other letter gets changed, then it's going to change it. And so it's not, we could get an incomplete protein or a faulty protein, one that doesn't function the way it's supposed to. So this can be harmful. Sometimes it has no benefit. And then occasionally you get those beneficial mutations where things actually advance and become more likely to survive because of that change in that protein. Whether that change in that one amino acid changes the color of your skin or changes the color of your eye or your hair, it may make you more survivable, and therefore your offspring would pass that mutation on to their offspring until it proliferated through the population. So here are some examples of issues that can be caused by point mutations, because sometimes they are not harmless. And that's it for the notes. So the assignment for today, Tuesday, was worksheet 7. 3rd hour finished this today. 7th hour did not. 7th hour is going to have class time Wednesday to finish this up. That's it for this video. It went a little longer than I wanted to. That's why I was kind of rushing there at the end, because you can always pause and go back. That's it for this. Final is on Friday. It will be posted as a Google form online so that those of you who are not here can do it. That's it. Have a wonderful night, and I look forward to seeing everybody, if not back in class tomorrow, maybe back in January. So, have a good one.